Okay, ladies and gentlemen, children of all ages, I'm about to go over some major microbiology information with you. We're going to be talking about Chapter 1, Chapter 3, and Chapter 10. Now, instead of going over loads and loads and loads of paperwork and quizzes and questions and 4.6 hours of teacher's lectures that I did, or doing fun things like crossword puzzles or going over fun graphs like that, or more quizzes that I took, or even doing fun crossword puzzles. We're not going to do any of that today. We're not even going to go over my stacks and stacks of notes that I do with fun pictures and colorful drawings. We're not going to do any of that. Why? Because we don't have time. We got a class to get to tomorrow. We have an exam. Instead, I'm going to tell you everything you need to know on the exam using this handy dandy 3x5 index card. Now, the teacher was nice enough to allow us to type it up. I'm extremely grateful for that because I have terrible handwriting. I have terrible spelling, which is why I do the crossword puzzle. And on top of that, I write big, possibly because I need glasses. Not so sure. Anyway, so I'm going to tell you about everything that you need to know on the exam with the 3x5 index card, not the stack of papers that I've been working on for the last, month that we've been doing this, okay? So we're going to go over some key concepts. The first one is oil. And I'm not talking about the type of oil that we're going to cook with later tonight. I'm talking about immersion oil. This type of oil gets used in when you're using a microscope. Now, why would you want to use the immersion oil? So I said here on my index card, and I kind of shortened it, so I'm going to give you a little bit more detail, is it's used to displace the air between the lens and the specimen. We use the oil to prevent refraction. So essentially what I'm trying to say is if you have your specimen on the nice little plate and you have your lens, what happens is there's all this air and the light goes poing, ping, ping. We don't want any of that. No, we want the light to go all the way through. So in order to do that, we're going to have, and I'm going to use this, we're going to put some oil on there, okay? And so what happens is that oil has the same refractory index as the lens, so therefore the light can go straight through, okay? It's not going to get lost in the air and refract. So what happens is it gives us a higher resolution, okay? So next, let's move on. So next, we might get some sort of question about an Acidfast Acidfast stain. So it's important to note why do we want to use an Acidfast, right? Because in lab last week we used crystal violet and methylene blue, okay? So an Acidfast is particularly used because we have like a endospore, okay? And some things like a mycolic acid, I believe is the name, that mycolic acid, the gram stain is harder to stick to. And so as a result, we need to use something that's a little bit harsher. No, it's called Mycobacterium, I believe is what the endospore is called. And it's a mycolic acid, that's the waxy substance that's around it. We'll get to that in more detail in a minute. But we're talking about Acidfast. So essentially, let's say we were to use the Acidfast stain on something that was that's actually gram stain. What would happen? What would happen is it would turn blue. Okay, because if it was If the Acidfast worked, then what happens is once you go through the sequence of steps, it would be red. But if it's not, in the case this would be a bacterium, it would be blue, okay? At first, this concept kind of tripped me up, but once I looked at the image, it was actually quite simple. So, moving on. Well, I put here it's blue because it's non-acid-fast. That's just the way it is. If the color is going to turn blue if it's non-acid-fast. It's going to turn, what color did I say? Red if it is in fact acid-fast. So that's all you need to know. If you have a gram positive, gram negative, you use acid-fast stain. It's going to show up blue because it's actually non-acid fast. All right. Next, let's say we have a weak cell wall and it's in a hypotonic solution. Hypo is big. Whoa. So we have this hypotonic solution. So what's going to happen? So on my little 3x5 index card, I said the cell pops, or it's called lysed, okay? Not like lice in your hair, but lysed as in pop. So this is because there's too much pressure on the cell wall. So essentially what's going to happen is water is going to get in, it's going to get in, it's going to get in, and that wall's going to get bigger, 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 and go poof, and pop, okay? So that's it. All right, moving on. So let's say you have a hypertonic solution, okay? And first, let's talk about what a hypertonic solution is, okay? So the example I'm going to give for this hypertonic solution is salt, okay? So let's say you got all this salt water, you know, swimming around in this pool of salt water, and then you get a cell. So here I am, I'm the cell, and I'm sitting in that salt water, right? And I'm hanging out, and I'm hanging out, and I'm hanging out. So what's going to happen is potassium is going... Shh, hush, Sassy. Potassium is going to come into that cell. It's going to go inside me. Whoa! Whoa! Now, why would I... Sassy, you're going to have to go outside if you do that. She wants to get in on the action. So, why would I, who's super lazy, right? I'm the cell being super lazy. Why do I want to spend so much energy trying to pump potassium in? Okay, I had to put her away. So why would we want to pump potassium in? Now, I got kind of hung up on this at first. So really, the big picture is, why is it going to pump an ion in? Okay, so I put here on my 3x5 index card, if the cell is placed in a hypertonic environment, it will use ATP, that's energy, guys, to transport ions into the cell to equalize the internal and external solute concentration and prevent plasmolysis, otherwise known as crenation. Okay, so essentially what's going to happen is you're in this hypertonic solution, okay, and what's going to happen is inside it's going to shrivel up, it's going to start pulling away and shriveling up. Well, it doesn't want to shrivel up because if you shrivel up, you're going to die, but I don't want to die, so I'm going to get some potassium and go pew, pew, pew in order to keep it from dying, okay? So, what's next? I put a little note in here if it's a hypotonic solution. What's going to happen is, so see, water goes from a high concentration to a low concentration. So if a cell gets put into a bunch of water, okay, the high concentration of water is going to go inside that low concentration of water, and the cell's going to swell, otherwise known as lysed, or I say popped, okay? Next, let's say you inhibit the growth of, so essentially there was a question, and the question talked about you want to stop the growth of a bacteria, but as a result, something's going to happen to the animal cell. And I'll get a little bit more into this with what I put. So you have a prokaryote, which has something called 70S ribosomes, okay? And eukaryotes, although mostly they have 80S ribosomes, they have 70S in their mitochondria. What? They have 70S in their mitochondria and prokaryotes have 70S? Why could this be? They think it has to do with the endosymbiotic theory, which we'll get to that in a minute. So we have the prokaryotes that have 70S. That eukaryote mitochondria has 70S. So if you destroy the 70S in your prokaryote, you destroy it in the eukaryote. So let's say here's the bacteria, okay? Bacteria has 70S in its ribosomes, and it gets inside of me. It's inside, okay? You destroy that 70S prokaryote. Well, in my mitochondria, I have 70S. So if you destroy that, you're going to destroy me, because 70S ribosomes that are in the mitochondria give me energy. Without that if you destroy that 70S in that mitochondria, I got no energy. I got no energy to go to class. I got no energy to tell you about my index card. I got nothing. Then what's going to happen is slowly and surely I'm going to die. So you can't go after that 70S ribosome, because you go after that 70S ribosome in the bacteria, you're going to go after the animal. That's a bad gig. Okay. So let's see. All right, moving on. So let's say you have penicillin. Penicillin. What about penicillin? Penicillin inhibits the peptobridges. Now, I know I said that I wasn't going to break out my other piece of paper, but I think if I didn't, you really wouldn't understand the concept, okay? So the teacher was nice enough to hand us out this little peptoglycan sheet, okay? And at first I was like, I don't need that. I got a book with colorful pictures in it, right? But then I was like, hold on. That sheet can be pretty useful, because I can draw on it, I can color it, I can even use it in a video. So thanks, Teach. So we're going to talk, here's our little image right here, and we have penicillin. So penicillin inhibits a peptide bridge. So what is a peptide bridge? So if you notice how these two, 1, 2, 3, 4 means tetra peptide. But see, you see how these two peptides right here are connecting? There's actually an inner bridge in there, and that's called a peptide. What is that called? A peptide bridge. So penicillin inhibits the peptide bridges. So it will inhibit the bridge that connects these two pieces. And if it inhibits the synthesis of that bridge, it inhibits the formation during peptidoglycan synthesis. So essentially, peptoglycan kind of blocks the synthesis of penicillin kind of blocks the synthesis of the peptoglycans. Kind of more simplistic way to say. But really, for that test tomorrow, you're really going to need to say what. So the penicillin is preventing the synthesis of that peptide bridge that's holding the tetrapeptides together. And actually, if you want to get even further, so you see how that little tetrapeptide is connected to that little ball right there? That little ball is called NAM. So only those tetrapeptides are connected to the NAM. We'll get to that later. All right, so what else does my little handy dandy 3x5 index card say? So, bacteria have peptidoglycogen, but archaea do not. Why? See, bacteria have peptidoglycogen. Archaea do not, and eukaryotes do not. Archaea actually have something called a pseudopeptidoglycan, but we're not going to get to that on our index card for the sake of time. And the teacher's probably not going to ask that tomorrow. So, bacteria have peptidoglycan, but archaea, do not. Therefore, the penicillin would not be presumed to work on the inhibiting growth of archaea. All right, so does that make sense? So, essentially, bacteria have the peptidoglycan, the archaea do not. Therefore, penicillin would not be presumed to inhibit the growth of archaea. It's not going to inhibit the growth of archaea because archaea doesn't have the peptidoglycogen. All right, moving on. Gram positive, gram negative, and medication. Specifically, the question was asking about gram negative and how that relates to medicine. Okay? So, in gram negative, first to start off, gram negative is resistant to antibiotics. That's a big key. Secondly, remember how we were talking about peptidoglycogen? Well, in gram negative, it has a thin layer of that, and it's sandwiched in between, so it's sandwiched in between two membranes, okay? Those membranes protect that peptidoglycogen, okay? So that's one reason why it's antibiotic resistant is because that peptidoglycogen is sandwiched in between the membrane, okay? You know what? I just realized why I got something wrong on a piece of homework. Anyway, learning stuff. So next, the polysaccharide, the lipid polysaccharide. What is a lipopolysaccharide? That's crazy. How am I going to explain this? So you have lipopolysaccharide, and that has something called antigen. Oh, let me not be dyslexic for my video. Lipid A and O antigen. So lipid A anchors. A anchors into that membrane, okay? So let's say this is lipid A. What happens is then the O antigen, this is my O antigen, hangs out the top, okay? And the whole thing is the lipid polysaccharide. So you have this anchor, this let me make sure I don't get backwards. You can't tell I'm a little bit dyslexic. So you have lipid A that anchors, and you have the O antigen. And so the lipid A anchors the LPS, like I said, right? And it's responsible, the lipid A is responsible for an endotoxin. I found it unique that this lipid A, really what it's doing, it's kind of setting off this immune response within us. It's not necessarily poisoning us, but our immune system is like, oh, holy moly, we got lipid A. So it sees it as an endotoxin, and it sets off this immune response. Then you have antigen O. Oh my, we have an antigen. So what does that mean? Antigen O is a second important medical thing to talk about because it's important to know the strain. So the O antigen, you can have different strains, and so you want to know what strain it is in order to know how to treat it, okay? So the other little bits on my card that we're going to talk about is emergent diseases. Why is this happening? So emergent diseases is happening, they say it's because of vaccinations. We all know I adamantly disagree with that based on over 300 hours of reading. but the book says it, so therefore we're going to put it on the quiz tomorrow, or the essay, the exam tomorrow. Okay, so if it's an essay question, you better put down that vaccine, that lack of vaccinations is causing this, regardless of what you believe. Next is old people. What does that have to do with emergent diseases? Well, when you're old, you don't have that great of immune system, okay? And so therefore you can get sicker. I would speculate that it's not that being old has a smaller part to do with it, but that it more has to do with the fact that older people aren't getting like booster shots and things of that nature, and so if you're not up to date with that, I don't know. I have a bunch of different things about it, but we're not going to go into it. So let's just stay on point. Emergent diseases are happening because lack of vaccinations, old people's immune responses, and bacteria resistant. Definitely agree with the bacteria resistant. Okay. A side note, there's a guy named Woes. Whoa, what did he do? He's the one that came up with the new categorizing of domains. He decided to do take prokaryotic and put it into two different groups, the bacteria and the archaea, and then don't forget over here was the eukaryote. He's the one that used DNA, the ribosomal DNA, in order to sort all of that out. And Kahn had to do, he was the one who actually figured out about endospores. See, I thought that was Tyndall, but Tyndall actually didn't do that. Tyndall just realized that there was heat resistant. It was Kahn that came along and said, hey Tyndall, thanks for giving me the input about heat resistance, but hey, did you know what it is? It's endospores. So that's what he did. The other guy that we can talk about, and this may be on the exam tomorrow, is about Louis Pasteur. He was on a Seinfeld episode, a Frasier episode the other day. They baked a cake with Louis Pasteur, and I actually knew who he was. So Louis Pasteur, what did he do? So he had these swan neck, these glass, kind of like these glass balls, and then it had the swan neck. And so what he did was he put broth into, they called it a swan neck, I think he called it a swan neck flask. But so he put it in the swan neck, and he took the broth and he sterilized it. And so what was unique about the swan neck is it allowed air to go out and it allowed air to come in, okay? The other thing that was interesting is that, so he let the broth cool and air enter, like I said. But see, because of that little bend, there was this bend in the swan neck. Because of gravity, the bacteria settled in that little bend. So that was very important. And then he let this sit for years. How the heck? He must not have had any kids, because I swear, if there was kids, somebody would have come along and knocked over his little experiment. But he was able to leave this thing sitting there undisturbed for years, and nothing grew, right? How could this be? Well, it's because of the way that he did that little jar in the swan neck thing, right? So the broth stayed sterile. Then what he did was he tilted it. And so it allowed bacteria to come into contact with the broth. So the broth and the bacteria came into contact with each other. And then he left this for hours or even days. And then microorganisms multiplied in the broth. So what was the whole point of this experiment? He wanted to say life comes from life. He didn't say it's called biogenesis, life from life, right? Whereas before, people had this theory of spontaneous generation where life just pops up from non-living things. So he wanted to figure out what was going on, and this experiment helped prove that. Now, it's important to talk about here just for a second that there were other scientists like Needham hammed it up and messed it up. There was like Lazzaro, I believe, Pasolini, some crazy name like that. And they did all of these experiments, and some of them could replicate, some of them couldn't replicate. And so essentially after 200 years of these people, you know, kind of knocking around all of this stuff, they finally figured out, well, it's because one, they're not heating the broth long enough, or two, they're using stuff like hay, and then there's like endospores that's not being killed off, so then that's going to create all kinds of bacteria and everything. So essentially that's what's going on with that. So next, we're going to go to this side of the card, but let's take a break.